I'm the secretary for this year's UCC Translational Medicine Society. So I would like to welcome you all to our next speaker, Professor Mark Ledwidge, and just as well to say a massive thank you to Mark for being so enthusiastic and so helpful and helping really realising this conference. So Mark is the co-founder and the director of Heartbeat Trust, Solvatron Therapeutics, LifeLab Therapeutics, and the Adatom Group. So Mark's work across all ventures really does epitomise that of translational medicine and the concept of bringing ideas from bench to bedside and essentially advancing human health. So I will now pass you on to Mark, who will be speaking about the topic of genomics in heart failure therapeutics.
Thanks, Ava, and thanks to everybody, and well done to you guys for putting together such a great conference. Hopefully next year's one we can have a little bit more in person. So if you can see my slides here, I'm introducing to you Tom and Mary, and Tom was 42 years of age when a cardiologist diagnosed blocked coronary arteries and a very high risk of heart failure. And Tom's own father had died young of heart failure. And this is likely hereditary because these are my parents actually, and it turns out that I have similar issues. And the good news for everybody in our family is that, as you can see here, Tom is now in his 80s. He's doing great, and he's truly beaten his genetic risk for heart failure with many medications, a little bit of surgery, and lots of golf. Now, really in cardiology, we've always set a lot of store by a family history. And I'm going to tell you a little bit about how the group that I work with is trying to meet the challenges of people with heart failure, and even people like Tom and Mary, especially using genomics. Now, why does this really matter? Well, it turns out it's a common story. Simply put, heart failure is where the heart doesn't provide enough blood to meet the body's needs. And about 1 in 5 people in Ireland of Tom and Mary's age have the condition with quite severe symptoms. And in the middle graph, in terms of incidence, I mean, the figures are difficult to actually pin down accurately, particularly in Ireland. We're probably better than the US, but not as good as some of our European colleagues. But one thing is for absolute certain, and that is that along with rising levels of obesity and diabetes, and also as we live longer, particularly in places like Ireland, where the numbers over the age of 65 are going to triple in the next 20 years, heart failure is going to increase a lot. Now, we also have challenges with the process of creating medicines in heart failure. And from the first account of using digoxin and foxglove leaves about 250 years ago by a guy called William Withering, to the discovery of the ACE inhibitory protein in the early 80s from the saliva of the pit viper snake, the process that we've used by and large in discovering medicines for heart failure, and in fact most medicines, is a sort of a forward pharmacology approach, let's call it that. And what that means is taking chemicals from nature and putting them into medicines and then putting them into good use. And there are at least 3 problems with this type of approach. First, let me see here we go, yeah. So first, we have a finite number of chemical entities. And secondly, and particularly in heart failure, you can see that the process takes too long, between 25 and 45 years for the first 3 disease-modifying therapies in heart failure. And thirdly, we've got a lot of late-stage phase 3 clinical failures, and this is a real challenge for us. And nowhere is this more acutely felt than in heart failure. And also the reasons why drugs fail is changing, the profile. And you can see maybe about 30 years ago, there was an even 3-way split between efficacy problems in orange, problems getting the drug to the right target, distribution, metabolism, kinetics, and that's in blue, and then toxicity, safety, and other issues in the rest. But now, the main reason why drugs fail is actually clinical stage efficacy failures, and often that happens in phase 3, and this is the most expensive, almost the final hurdle of the drug development, and that has huge public health implications. And this is best exemplified by the form of heart failure where the pump is okay, but the heart is still not functioning well. And we call this heart failure with preserved ejection fraction, or HFpEF. And I'm going to use that term a little bit, so I'll explain it here. So very, very simply, as you can see in this graphic, when the heart is pumping out blood or in between heartbeats, it must relax and it must fill with blood before the next pump, before the next heartbeat. And if the pump function is working fine, but the heart is not filling with blood, it still cannot perform properly. And this is what we call HFpEF. And in particular, we have great therapies for heart failure where the pump function is a problem, but systematic evaluation of the 5 therapies that are disease modifying in heart failure with reduced ejection fraction, you can see them written down here, have failed systematically over the last 20 years in this form of heart failure with preserved ejection fraction, or HFpEF. And this is a real public health concern, because right now, HFpEF is actually the major form of the disease, about 6 in 10 people, and it also affects older people and women more than men. So we need new strategies, a little bit like Anthony Fauci and his new boss, Joe Biden, in tackling the coronavirus and pandemic in the last couple of weeks. And I'm sure you've been hearing a lot about that today. And sometimes the best new strategy is sort of the oldest and most obvious one. If you can't cure a condition, well, then prevent it. And Ken MacDonald, myself, and a fantastic team, the STOP-HF team, initiated the St. Vincent's Screening to Prevent Heart Failure study, or the STOP-HF study, more than 15 years ago. And we recruited people with normal ejection fraction, normal pump function, and risk factors for HFpEF, diabetes, obesity, and hypertension. But they were otherwise well. And we used a blood test called B-type natriuretic peptide, followed by heart imaging with Doppler echocardiography or ultrasound, to stratify patients into 3 stages of heart failure. Stage A, where they have some risk factors, but no symptoms and no sign of heart damage on the ultrasound. Stage B, where the patients feel fine, there's no symptoms, but there is damage to the heart, and we can only see that when we use ultrasound. It's below the level of the water, if you like, it's below the waterline. And what we're trying to do in this process is we're trying to prevent people going on to develop the more severe symptomatic stage C heart failure, where the symptoms and the signs are much more obvious. What's unusual about the study in many ways is, apart from the clinical characterization and the biochemical characterization using blood tests, and also the collection of lots of details about their medicines, we do Doppler echocardiography in everybody, including the stage A patients. And while this is mandated by some guidelines, for example, for people with hypertension, it's rarely done in practice in Ireland, or in fact anywhere in the world. So this is an unusual group of people. And because we follow them up for such a long period of time, we can evaluate the impact of this biomarker-driven strategy. And initially, we did it in a randomized controlled way. And you can see in the middle graph here that we show that there was a reduction of more than 40% in the development of heart failure. And also in the bar chart on the right here, you can see that patients also had 40% reduction in lots of other cardiovascular events. So this strategy has become the standard of care in our unit. Other people have tried to replicate this successfully, and now it's been incorporated since 2017, into North American, Canadian, and other international guidelines. And using BNP is quite a good idea, it turns out, because BNP is a cardiac hormone. It's produced by the heart when it's trying to protect itself from stress and strain, and also inflammation, fibroinflammation. And you won't see the detail here, but the heat map on the screen shows that BNP is very highly expressed, particularly in the upper chamber of the heart, which responds to the pressure of a stiff heart. So it makes sense that this would be a good blood biomarker and a good blood test to use for risk stratifying patients. But what we're now trying to see is, well, would it actually help us to use genetic risk scores in our population to also help risk stratify? And what I'm showing you here are unpublished data. These are new data from our unit using a genetic risk score of damage to that upper chamber of the heart on top of BNP. And the people on the very right in the gray bar have a 4 in 10 chance of developing heart failure over a relatively short 5-year follow-up period. And this is independently twice the level of people with the lower quintile of genetic risk, but an elevated BNP. And we can't really tell the difference between these high BNP with low genetic risk score and high BNP and high genetic risk score people from the way they present in the clinic. So there's definitely some potential use here. Now, we need to do a lot more work on this. I mean, first of all, we need to validate this work in our own population and probably other populations. And then we need to figure out how to communicate this risk to our patients so that it's beneficial and it's useful and it's actionable. But it certainly shows that there is a huge potential here to improve the risk stratification and improve prevention, hopefully, of heart failure. The second strategy that we've adopted in our unit is to try and focus on disease subtypes, recognizing that, you know, even within the umbrella of HFpEF, one size doesn't fit all. We don't have the same patient right across the spectrum. And some very interesting work by a collaborator of ours, Sanjeev Shah from Northwestern University in Chicago, published a few years back, where he used machine learning on the clinical characteristics, some of the biochemistry and BNP, and also on the imaging characteristics of paper. He used the machine learning to independently or in an unsupervised way pick out the subsets in a way that maybe it's harder for humans to do. But what it showed was something that made sense to clinicians. It picked out at least 3 subtypes of this group. The first group, they tend to be younger. They have very, very early stage mild dysfunction and very low levels of BNP elevation. The second group, they're maybe a little bit older. They tend to have diabetes and they tend to be obese and have sleep apnea problems. And they have an intermediate level of risk, you can see in the bar chart on the left here. And then the third group, these are older, they have chronic kidney disease, they have pathology in the upper chamber of the heart. It's usually expanded, it's usually bigger, it may have atrial fibrillation, and they also have very high levels of BNP. And for us, using BNP to potentially prevent problems in this subtype 3 became attractive a few years back. And when we initially suggested it, not everybody was convinced this is a good idea. BNP is a protective hormone, and that's our view of things. And that way, it's not like cholesterol, for example, where the level is high, and that means there's a risk. And so what you want to do is drive it down. With BNP, the level is high, that means there's risk, but maybe what you want to do, because it's protective, is boost it up. And with some colleagues in the Mayo Clinic, we genotyped the population, especially focusing on this subtype 3 patient group with HFpEF, for a promoter region variant on the BNP gene. And this promoter region variant G allele, which is heterozygous in the middle bar, or homozygous on the far right bar, where you've got 2 copies of this BNP boosting allele, you can see that by the time that patients arrive to us in their 70s, they already have a much lower, a significantly lower risk of hypertension. And when we followed these patients up for the next 5 years, we found that those GG allele patients, even though they had 40% higher BNP, had much less progression to heart failure or the substrate for heart failure. So this gave us the confidence to invest our time and the team's time and our patients' care into the Parable study. And we began this about 5 years ago. We had last patient, last visit in December 2020. I know that Fiona Ryan is online here. Fiona Ryan has to take a huge amount of credit for helping myself and Ken run this project in an unbelievably professional way. So thank you to Fiona on that. And this actually is going to help us understand whether pharmacologically boosting BNP, because we can do it with a drug called sacubitril valsartan, will actually help prevent progression of heart failure. We haven't unblinded the data yet. We're still doing data QC, but we hope to report on this in the next couple of months. But what is interesting to us is that the event rate in this population, as we would predict for these stage 3 patients, is about 4 times higher of the general cardiovascular or the general STOP-HF cohort. So, and this also actually, we're using cardiac MRI in this study, and it's one of the largest studies in cardiac MRI that's been done to date. So genotyping helped us have confidence to invest in Parable. And it turns out that lots of people are doing this. And this study published 5 years ago in Nature Genetics showed that when there were genetic or genotyping evidence or genetic evidence to support drug development, that in general, it was twice as likely to be successful. And in particular, if the effect size or associated with the gene or the gene variant was high, and what I'm talking about there is an effect size of greater than maybe about 3 or 3.5, there was an up to 5-time chance of success. So this is really, I think, a very important part of the future for drug development. But that's about genes and variants that we already know. What about new targets? Well, I think in many ways we're really at the kind of cusp of something very interesting here. We haven't quite got it right yet, but in heart failure, the largest genome-wide association study with 46,000 patients was published in 2020 in Nature Communications. And while it defined lots and lots of targets and lots of associates of heart failure, unfortunately, they were all targets that we knew about. It didn't reveal any new cardiac biology. And this is a particular problem in general for developing drugs, because year on year over the past 3 decades, the number of new targets coming to the market to help treat patients globally is in decline, about 10% on average decline per annum. And also, while we know that there's somewhere around 19 or 20,000 protein coding genes in the body, we only actually have defined drug targets in the clinic for less than about 5% of them. So there's a lot more work to be done. Now, the question for me is the genotyping strategy the right way to go? I mean, genotyping for large groups of patients it's affordable, it's feasible. But the problem is, a little bit like that Hermes study, there are lots of gaps. And new data, derived from the UK Biobank last year and the year before, shows that what these tend to do, genotyping studies tend to do, is tell us stuff that we already know. It doesn't tell us about new variants. It doesn't reveal new biology as easily, and particularly when the variants are low frequency. And if you were a betting person, you would say that the highest impact variants, the ones that we really want to look for, they're going to be low frequency. They're going to be rarer potentially. And those are the ones that we want to try and find. Genotyping isn't going to do it. What we actually need to do, we think, is whole genome sequencing. And so this is the most, and I'll try and finish up soon because I know you maybe want to have some questions on this. This is probably the most exciting thing that I've been involved in, in my career. And this is a whole genome project to genotype about 12,500 patients with risk of heart failure, but also with heart failure. And we're going to enrich this for HFpEF so that we have the ability to drill down into these subtypes of HFpEF. This is a huge collaborative effort that is involving a number of units around Ireland. We've started recruiting into this study. It's also including our STOP-HF program. We've pulled together a group of all of the people that Ken and I have worked with over the last 20 years internationally, a really great bunch of people internationally to help us doing this, particularly from North America. And Genuity Science are providing all of the whole genome sequencing resource. I mean, the cost of whole genome sequencing at a very high quality is somewhere between 800 and 1,000 euros per patient. And that's before you do any work with the data or store the data or mine it for new ideas about where to develop the next generation of therapeutics. And we've whole genome sequenced about 3,500 patients so far in this project. It's already the largest whole genome sequencing project, I think, in heart failure. And we have over 5,000 controls. And it's a real multidisciplinary effort, you know, cardiologists, pharmacists, researchers, nurses, lab scientists, GPs on our side, on the Stop HF side, but also then geneticists, bioinformaticians, computer scientists, artificial intelligence experts, and statisticians on the genuity science side. We're going to look at multiplex biomarkers to try and make connections between proteins and these genes. The workflow is complex, but, and I won't get into the detail here, maybe we can talk about it over questions, but what I want to tell you, because I'm quite excited about this, is that already we've identified 2 new low frequency, high impact variants from this research that have never been associated with heart failure before. And this is particularly associated with our subtype 3 heart failure. Remember that group of people that are probably faring worst with HFpEF? And the odds ratio, or the effect size associated with these 2 new variants, is 10 and 11. So these are low frequency, much less than 2% for both of them, but also very high effect size. And we're now beginning to start the process of figuring out, you know, where would we begin to validate and to maybe understand how these variants could help. So back to Tom and Mary, back to my parents. So Tom has been involved in all of the projects that I've told you about before. Like many of our patients, they give selflessly of their time because they want to be involved in the research, and they want to be involved in helping us, you know, improve screening and prevention by maybe down the line adding genetic risks, avoiding this one-size-fits-all approach, and maybe using genotyping to support the work that we might do in investing in expensive clinical trials like Parable, and finally in using whole genome sequencing to hopefully find maybe the first disease-modifying therapy for HFpEF. So thanks very much.
Brilliant, Mark. Thank you so much for that. Incredibly insightful. And I just think it really does go to show that patient impact makes such a large difference in these strategies. And I know that when I first met you, when you mentioned about the HFpEF, and that it was kind of a concept that was overlooked in heart failure compared to the, you know, reduced ejection heart failure. And I think it's something that your work is really filling the gap in, and I think that is really incredible, and that we've been able to see some real data of that today. So I thank you for that.
Great, thank you very much.
So we might get on to a few questions, if you don't mind. So I have one here. Is what, in your opinion, is the most profound challenge facing the development of therapeutic targets currently, be it through lack of communication between clinicians and researchers, etc.?
That's a great question. I think the biggest issue is time. I think we waste a lot of time, and I think most of that time wasting can be addressed. I mean, we've got to have process, okay? We've got to validate what we think, we've got to replicate what we think, and that's going to take time. But we do waste time, I think, and I do agree with the premise of the question that, you know, more communication between the disciplines, between patients, between clinicians, and between researchers, and particularly now data analytics experts, I think is going to help reduce those timeframes. Brilliant,
brilliant. And I think that's probably what the aim of this conference is today, is kind of tackling these challenges in translational medicine and hoping to improve patient outcome. So thank you for that. And then I have another great question here. As someone living with heart failure, it is great to hear your work, very exciting. Do you see an increased numbers of genetic variants associated with cardiomyopathies concomitant with other diseases such as neuromuscular? Thank you.
That's another great question. And honestly, we don't have the answer to that. And I think, I mean, we do know that there are some fairly rare gene variants that are associated with a kind of genetic, they're called monogenic diseases, genetic cardiomyopathies. The association of those with other, you know, diseases is, you know, genetically is hard for me to say because we just don't have enough work on it. But what I do know is that heart failure is often, often presents to us with lots of other conditions. So it's very likely that that's the case. I think that really, I feel like we're at the beginning of a very important journey here in trying to understand the answer to the questions that person has just posed. And I think it's, you know, there's a lot of responsibility on those of us who have the interest and the ability and the team around us, because you can see it takes a lot of people to pull that together, to really work hard to try and find the answer to that question. And I think that the big learning for me in the last 3-4 years is just the sheer volumes of data that we have to rely on. And, you know, we can't store these data in the usual way in the hospital that we would have liked to. We have to store them in the cloud. We have to interrogate them with special tools that also sit in the cloud. And we need more dialogue between computer scientists, bioinformaticians to actually deliver on finding out the answer to the question that person has just posed.
Yeah, I think it's a great point, and I think there is massive scope for AI and computers and all of that in the future of translational medicine. So I just have one personal question. So if you had one piece of advice to students looking to pursue a career in the area that you're in now, or say drug development in general, what would you give them?
I would say be curious. It's so easy when you're you know, trained in a particular discipline to get very focused on, like for me, I'm a pharmacist by training. It's very easy for me to get totally focused and totally absorbed in that. But I think, you know, be curious about other people and what they're doing, because sociology, psychology, computer science, mathematics, physics, they're all as important to me in the work I do now as pharmacy. And I think that most people that work in our group would probably agree that you need to have that broad range of skills, and you need to be interested and appreciative of the skills that other people have. I think so that would be curious about your friends and what they're into.
Brilliant. Thank you for that. Very insightful. And I think that's kind of all we have time for in terms of questions. So I'd just like to, again, Mark, say a massive thank you for all your enthusiasm and your help thus far, and for such an interesting talk that I hope that you'll have great luck with in the future.
Thank you very much, Ava, and well done to you all.